Hello, this is Dr. Farhad Ravandi from the University of Texas MD Anderson Cancer Center in Houston. Welcome to this educational discussion on the role of antibody-based therapy in acute myeloid leukemia, during which we will explore what antibody therapy has meant historically in AML and also assess the evidence on a new generation of antibodies in clinical development. Joining me in this discussion today is my colleague, Dr. Amir Fathi from Mass General Hospital in Boston.
Thank you, Farhad. I'm happy to be here and I look forward to the discussion.
The following audio is from a two-part discussion with Dr. Farhad Ravandi and Dr. Amir Fathi. This audio is part of a certified educational activity titled Expert Discussions on Antibody-Based Therapy in AML. Will new technologies make a difference in patient care? To access the entire activity and complete the post-test, please go online to www.peerviewpress.com/NYX. A printable monograph, slides, practice aids, and other features are also available.
Let's begin by asking the question, why do we need antibody therapy in AML, and looking at what historically antibody therapy has meant in this setting. Antibody-based therapy has been of a lot of interest in various hematological cancers over the last couple of decades. In lymphoma, multiple myeloma, chronic lymphocytic leukemia, as well as more recently, acute lymphoblastic leukemia, there have been a number of antibody-based agents that have been approved for treating patients. Although we did have gemtuzumab ozogamicin approved in 2000 on an accelerated approval basis for treating patients with AML, this was withdrawn in 2010. Treatment of patients with AML has changed very little over the last several decades, and we are continuing to use traditional cytotoxic agents, cytarabine and anthracyclines. This means that we do need newer, more effective strategies to treat our patients in order to improve the outcomes of the patients in the near future. There are other antigens in AML that are of potential interest, as well as CD33 and CD123, and these are CLL1, CD47, and CD45. As I mentioned, gemtuzumab ozogamicin, a first anti-CD33-based antibody, was available but has been withdrawn, but more recently, vedastuximab telurin has been evaluated in a number of clinical studies that are being conducted. Next, we are going to discuss some of the anti-CD33 antibodies that have been developed in AML and take a look at how they target leukemia cells. We will also review some of the clinical safety and efficacy evidence in various AML populations. Amir, what can you tell us about CD33 as a target in AML?
CD33 is an attractive target for antibody-based therapy in AML. It is a cyclic receptor. It stands for sialic acid binding immunoglobulin like lectin. These are receptors that are seen in a variety of cells of hematologic origin. The expression of CD33 increases with myeloid maturation, and in general, it confers a negative regulation on activated cytokine receptors, among other features. Upon engagement by the sialic acid ligand, it undergoes endocytosis once it's activated. CD33 has the advantage of being expressed on the majority of leukemic blasts in AML, although their expression is variable. The question of whether it also exists on the leukemic stem cell is currently under significant investigation.
Over the years, there have been several attempts at developing antibodies that target the CD33 antigen. As we noted earlier, the most extensively studied have been gemtuzumab ozogamicin and vedastuximab telurin. Let us find out more about these agents. Amir, let's start with gemtuzumab.
Gemtuzumab ozogamicin is an anti-CD33 antibody conjugated to calicheamicin, which is a cytotoxic antibiotic. Calicheamicin, upon binding to the receptor CD33, is hydrolyzed in lysosomes after internalization. As Farhad mentioned earlier, it gained accelerated approval by the FDA in 2000 on the basis of phase 2 clinical trials, which showed an impressive response rate in relapsed AML. Soon after approval, increasing signals for liver toxicity in certain subpopulations started to emerge, raising some concern for the drug. And ultimately, given an increased rate of mortality in a randomized chemotherapy combination study, the drug was withdrawn from the market voluntarily in 2010. However, since that time, there have been a series of clinical trials looking at multiple patient populations with gemtuzumab ozogamicin in combination with chemotherapy, including younger patients, older patients, those individuals who have favorable risk AML, again, across a whole series of clinical trials showing advantages in survival and other outcomes, raising some hope that perhaps there is a future for gemtuzumab in AML.
Thank you, Amir. As you know, the experience we have with gemtuzumab ozogamicin has informed that the search for newer CD30 targeting antibodies could be fruitful. What are your thoughts on the development of vadetuximab taurine, one of the newer generation CD33 antibodies?
Well, I believe this is a very exciting agent, an exciting antibody drug conjugate. It also is a CD33 targeting compound. It is coupled to another cytotoxic drug called PBD, pyrrolobenzodiazepine dimer, which cross-links DNA. It has a very stable cross-link. It's less affected pharmacokinetically and by P-glycoprotein. The compound is an anti-CD33 antibody, which upon binding of the receptor is internalized, with the complex then being trafficked by lysosomes, and thereafter the cytotoxic agent is released in the cell, binds DNA, impacts DNA repair, and thereafter promotes apoptotic cell death in a targeted fashion. In monotherapy studies in predominantly relapsed refractory populations, vadetuximab has demonstrated anti-leukemic activity in a significant proportion of patients. Among those who received the dose of 40 micrograms per kilogram, which was an extensively studied dose in the monotherapy study, 47% achieved blast clearance. The overall composite remission rate was 27% across the range of doses that were studied in the monotherapy trial. A large proportion of patients achieved significant blast clearance on study, more so at higher doses of the antibody drug conjugate. And many of those patients who achieved blast clearance also achieved disease response. There were some toxicities related to vadetuximab taurine, predominantly on-target effects, myelosuppressive effects, which were characterized with mainly cytopenias, such as thrombocytopenia, anemia, neutropenia, febrile neutropenia. However, 30-day mortality was low at 6%, and the recommended monotherapy dose of 40 micrograms per kilogram was identified based on this initial data. There was some rationale for combining hypomethylating agents with vadetuximab. In preclinical studies, hypomethylating therapies appeared to increase modestly the density of CD33 on the surface of leukemic blasts. In addition, it appeared that exposure to hypomethylating therapy seemed to increase the incorporation of the cytotoxic agent, PBD, into the DNA of leukemic cells. This rationale for potential synergy between hypomethylating therapy and vadetuximab taurine then led to a cohort of the phase 1 study looking specifically at the combination of hypomethylating agents with this antibody drug conjugate in newly diagnosed patients who were not otherwise eligible for aggressive induction chemotherapy.
Now that we have reviewed some of the evidence with vadetuximab in AML, let's take a look at some of the recent updates from the ASH meeting in December 2016 in San Diego. Amir, what is your take on the update on this agent?
There have been some exciting studies presented at this year's ASH meeting. The combination of vadetuximab taurine with HMAs was updated in an oral presentation. The combination continues to be well tolerated. There is no identified pattern of off-target toxicity. There is improved activity compared with historical HMA monotherapy in older AML patients. The composite remission rate is above 70% in this patient population of predominantly older patients. The majority of patients on this cohort of the study were above the age of 75, which is remarkable. A significant number of patients had poor risk features such as high-risk karyotypes, and therefore this result was deemed to be quite promising. activity was maintained across the highest risk patient groups, including patients over the age of 75. Vadetuximab has also been studied in combination with chemotherapy, traditional cytotoxic chemotherapy, such as 7+3 induction therapy, as well as with consolidation and maintenance approaches. In an abstract presented by Dr. Erba and colleagues, the combination of 7 and 3 plus vadetuximab talarine was found to be well tolerated. There was an acceptable time to count recovery and a very high rate of remission, with the large majority of patients achieving MRD negative state, which is quite promising. Extramedullary adverse events were uncommon and similar to prior reports of 7 and 3 alone in this AML population.
Vadetuximab is now being tested in a phase 3 randomized trial in newly diagnosed patients with AML. Adult patients who are not candidates for intensive cytotoxic chemotherapy are eligible to participate. The randomization is between hypomethylating agents alone or in combination with vadetuximab talarine. The choice of hypomethylating agent could be either azacitidine or decitabine, depending on the investigator's choice. Thank you, Amir. I think this was a very excellent recap of the data. I think it is clear that we have learned a great deal from gemtuzumab experience in AML, and this will be useful going forward as we continue to evaluate agents such as vadetuximab. In the next segment, we will explore some of the other new antibody-based approaches targeting CD33, as well as other new antigens that are being evaluated as targets for AML therapy. Welcome back. Let's move on and discuss other approaches targeting CD33, as well as antibodies that are being developed against different antigens, such as CD123 and CD47. Let's begin with lintuzumab radioconjugates. The actinium-225 lintuzumab is a radiometal that emits 4 alpha particles linked to an anti-CD33 antibody, limtuzumab. Fractionated dose actinium lintuzumab combined with low-dose Ara-C in a recent study in all the patients with untreated AML was evaluated, and it was shown to be combined with low-dose Ara-C reasonably safely. Antileukemic activity was observed with this combination. Another agent, AMG 330, is a bispecific T-cell engaging antibody with anti-CD33 activity that is currently being investigated in a phase 1 trial in relapse and refractory patients with AML. Amir, what do you think about the other compounds that are in development, particularly CD123 and CD47-based antibodies?
Leukemic resistance appears to be mediated by a fraction of malignant precursor cells in the marrow that are capable of self-renewal and are less sensitive to traditional chemotherapy. These leukemic stem cells oftentimes express CD123, which makes it a valuable target for therapy in AML. CD123, also known as interleukin-3 receptor alpha chain, is a key mediator of cell proliferation, differentiation, and survival. And although it is present on the large majority of leukemic stem cells, it is thought to be very low in expression on normal hematopoietic stem cells, providing some rationale to use the particular target in AML. Several other agents are currently under study that target CD123. These include HU707, a humanized unconjugated CD123 antibody, SGN-CD123, an anti-CD123 antibody drug conjugate, XmAb-CD3-CD123, which is a bispecific antibody, MGD006, which is an anti-CD123 dual affinity retargeting molecule, so-called DART compound, that combines CD123 targeting and brings the particular cell in close proximity to a CD3 expressing T cell. Other targets that are of note as for the purposes of treatment in AML include CD47, a cell surface protein regulator of phagocytosis. It is increasingly expressed in AML cells. Humanized anti-47 antibodies are currently under study. Additionally, CLL1 is another target on the surface of AML cells. It is an AML-associated stem cell marker. Its expression appears to be higher appears to be associated with a higher rate of relapse. Finally, MCCLA-1117 is another BITE compound that is under preclinical study.
Recently, there has also been an attempt to develop antibodies targeting CD45 to use as conditioning therapy prior to allogeneic stem cell transplant in AML. Amir, what do you think about these antibodies?
One such compound is IOMAB-B, a monoclonal antibody targeting CD45 conjugated to radioactive iodine-131. It offers a novel targeted approach to ablation as a conditioning compound prior to allogeneic stem cell transplant. An ongoing phase 2 study is demonstrating that IOMAB-B is safe and tolerable in combination with reduced intensity conditioning and associated with favorable overall survival in patients over the age of 50 with advanced myeloid neoplasms. The current phase 3 Sierra trial is seeking to look at patients with relapsed refractory AML over the age of 55, comparing IOMAB-B followed by stem cell transplant with physician's choice of chemotherapy followed by transplant.
Thank you, Amir, for a very interesting discussion today. I hope you have all enjoyed this overview of antibody-based therapy in AML. It is clear that there are multiple promising targets for antibody development, including CD33, CD123, CLL1, CD45, and CD47. There are a number of agents rapidly moving into phase 3 trials, including the antibody-drug conjugate vadetuximab, which has demonstrated activity as a single agent and in combination with hypomethylating agents and chemotherapy. In addition, we have several promising bispecific and radioable antibodies being tested in acute myeloid leukemia, which may offer us different ways of treating our patients with AML. I hope you have enjoyed this discussion and that you have found the evidence we have reviewed useful for your practice. Thank you again for joining us.
This activity has been jointly provided by Medical Learning Institute Incorporated and PVI, Peerview Institute for Medical Education. Thank you for listening to this activity. To view the rest of the CME activity, download materials, and complete the post-test for instant credit, please go online to www.peerviewpress.com/NYX. This activity is supported by an educational grant from Seattle Genetics.